Okay, hi everyone. My name is Adam Youssef. I'm also an NIH R15 fellow, and I worked with Dr. Staudinger and Dr. Kramer, as long as with Dan and Catherine over the summer at the MSSU KCU Research Consortium. And my title was The Interface Between Drug Metabolism and Inflammation in a Triple Negative Breast Cancer Cell Line. So which cell line did we use? We used the MDA MB231 cells. And there was a specific reason we use this. It's because, as actually mentioned before, it's a triple negative breast cancer cell line. And what does that mean? It means that out of the three key receptors that it could bind to or could get activated via, it doesn't get activated via those. So it makes clinically, it makes the cancer very difficult to treat. It also models late stage breast cancer. And our specific cell line expressed very low levels of pregnane X receptor, or PXR. And finally, it's known for the high inflammatory signaling, making them an excellent model for investigating how PXR agonism may influence pro-inflammatory gene expression in cancer. So looking at this figure over here, we see that PXR agonism by rifampicin is hypothesized to reduce inflammation via transrepression of NF-kappa B, which is a key transcription factor in the inflammatory response. And this would therefore blunt the inflammatory signaling cascade. Now notice here, LPS is what activates NF-kappa B, and we're gonna get into that later. So simply put, our objective for the study is we aim to determine whether activation of PXR blunts the inflammatory response, specifically in MDA MB231 cells. So this was a figure similar to student Dr. Russell's, and to touch on it briefly before moving on, we cloned human PXR cDNA into a stable expression vector, which we then used to generate our own PXR overexpressing MDA MB231 cells. Using this, we achieved something we called forced stable overexpression of PXR. And what that means is forced, well, we forced the overexpression of PXR, and stable, it remains stable throughout the study, which is what we achieved, or what we aim to achieve. Now, on this figure, on the left, we have a Kumasi blue staining. On the right, we have a Western blot. The Kumasi blue, simply put, just verifies the near equal loading across all targets of protein. So wild type, we have clone 1, 2, and 3, and I'll describe those later. And for the Western blotting, this shows the presence of the anti-PXR antibody in all clones, which is seen at clone 1, 2, and 3. Notice how we don't see any anti-PXR antibody in the wild type, and that is because the wild type has low or very low or no expression of PXR, and that's what we aim to achieve. So the genes studied over the summer were 6 main ones: IL-1 beta, IL-1 alpha, TNF alpha, MMP9, BAC3, and IL-6. So why did we choose to study all these 6 genes? We could have just chosen 1 or 2 or done something else. Well, all 6 genes are key mediators of inflammation. They're all pro-inflammatory genes. They're also all regulated by NF-kappa B, as we saw this figure before. Finally, it's a broad panel of genes, and this is something very interesting I learned over the summer. Pro-inflammatory genes don't just turn on and turn off, and then the anti-inflammatory section comes in. Pro-inflammatory genes have different timestamps and time points. There are early stage pro-inflammatory genes or late stage. So this is why we chose to focus on this broad panel. For this specific PowerPoint, though, just for time's sake, I'm going to focus on 3 of them: TNF alpha, IL-1 beta, and IL-6. Some nomenclature to be aware of: WT just means wild type. This is just a low PXR expression. And CL, it just means clone, which is PXR stable or stable overexpression of PXR. LPS stands for, and this is in the graph we saw, LPS stands for lipopolysaccharide. This is an endotoxin which exacerbates or induces inflammation. And R, as we will see in the graphs shortly, stands for rifampicin. This is an antibiotic. It suppresses inflammation. And an important thing to point out is the pretreatment with rifampicin. We pretreated these cells overnight or for 18 hours, as some studies have shown that treatment right away or not pretreatment does not agonize PXR to the same degree. So let's jump right into the results. So this is the first gene, TNF alpha. We're just focusing on the LPS, which, as we said, was the lipopolysaccharide endotoxin. As we see at the results, we're just comparing the lavender and the dark blue. The lavender has a huge fold induction, especially at timestamps 2 and 4. going to 40 to almost 55 fold induction. And this shows that overexpression of PXR in the clone cells induces inflammation in TNF-alpha. Now, before moving on to the next graph, the next graph includes all this data and the LPS and rifampicin. So it might seem like a lot, but I'll guide us through it. Let's just focus on the dark green and light green. So dark green is the clone with the LPS, the one we just saw in the last figure, and the light green is with the rifampicin. Notice at timestamps 2 and 4, we have a statistically significant decrease in inflammation, which shows that pretreatment with rifampicin decreases the inflammatory response. And again, this is TNF-alpha. So let's move on to IL-1 beta. Just right off the bat for LPS, we don't see any statistically significant differences. Everything was pretty much this comparable. Now moving on, it's going to include all the time points, but let's again focus on the dark green and the light green, excuse me. So in the light green, notice how at timestamps 2, 4, 6, 8, or actually 8 wasn't statistically significant, but also 10, we see a huge fold induction up to 20 or 25 fold when we pretreated with rifampicin. So this is something we didn't expect to see, and it's something that I'll discuss in the discussion section. And it just makes us think if there's something else going on, PXR independent effects that might be leading to these results. Finally, IL-6, as we see here, just LPS, again, the lipopolysaccharide, at 2, 4, 6, and 8, there's a statistically significant increase or difference, and it goes up, around 12-fold induction. And when we add in the rifampicin, it decreases the fold induction at time points 2 and 4, which is what we're expecting to see. But also look at time points 6, 8, and 10. We see a statistically significant increase in inflammation. And putting this all together, as a final results, we didn't see any blunting or uniform blunting of the inflammatory response. And actually, we saw some contradictory results, especially in IL-1 beta and IL-6. And so, as I said before, this might lead us to think that there's some possible PXR independent effects, things that might be going on in the background that we're just not 100% sure of currently. And further studies is definitely warranted to elucidate these results. These are my references, and I would just like to acknowledge all the people on this slide. The work would definitely not have been able to be accomplished without them. Thank you.
Oh, I think every student deserves a question. Hello.
That's a good question. I wasn't responsible for choosing the certain drug. I'm not sure if there are certain drugs. I'm not sure if there are certain drugs that, yeah, let's refer to.
So rifampicin is a well-known prototypical agonist of the receptor under study. Could have chosen some other ones, but for publication purposes, target audience would know immediately that rifampicin is prototypical agonist of that particular receptor.
Is this the same drug that's the aging drug too? We can have that conversation later, but yeah, it's in the literature in aging as well.
Rifampicin is an antibiotic that's used to treat meningitis and other things. So what is your question on that?
We don't want to hold this show up. A drug in the aging literature as well. It might be this drug. I'm not sure. Is it rapamycin? Yeah.
Yeah, you're thinking of rapamycin.
Okay.
Different.
Gotcha.